Yeah, I have a medical card too.
No, that's not working. It says it wants service.
All
All right. So, won't be even writing much on board, and then we'll get to 12 and 13, which is immune system. Then you're going to start hating me again. All right. So, we looked at the processes of the cells, all right? Replication, protein synthesis, cellular respiration. Okay, why are we doing this? Because with us, you have to first find, okay, what are the processes of the cells, how you are different from these pathogens, which are your enemies. So, for example, with bacteria, we realize that it is a prokaryote, so it is a totally different setup than us, a different setup of cells than us. And then the rest of them, like the fungi, protozoas, they were very similar to us. Helminths, the parasitic worms, again, they're eukaryote, their setup is okay. And viruses, they use our cells, right? So that's even totally something different. Same thing with prions. Why do we care? Is because you need to, if you're fighting an enemy, you need to know your enemy very well. You need to try to find differences between you and your enemy, so that you can target the enemy and damage the enemy without damaging yourself. So when we're talking about microbiology and infectious diseases, that's the purpose. Something has gotten inside and causing your abnormal physiology, causing you to get sick. So you want to target that pathogen and kill the pathogen, destroy the pathogen without adding to additional damage to your cells. So that's why we went through cellular biology and looking at the differences. So for example, the process of fermentation is a little bit different in prokaryotes versus us. When we come to DNA replication, not much of a difference. Helicase is helicase, DNA polymerase is DNA polymerase. So really, it's very hard to target it. Maybe we can use the sequence of DNA for, like we did with the PCR, for identification of the pathogen. And you can do it, it doesn't have to be a virus. We can do it with bacteria, anything like that, we can do a PCR. But toward the process of replication, really, like prokaryote gyrase was the only little tiny difference. But for eukaryotes, we're all similar. When it comes to protein synthesis, we can actually target the bacteria. They have a 7S ribosome, so their ribosome is different. We can target that. We can have the process of protein synthesis. But again, when it comes to eukaryotes, we're very similar. We have to look at differences. Structure of the cells. Peptidoglycan. Bombard that thing to hell. Nothing's going to happen to you. Why? Because you don't even have anything that looks like peptidoglycan. So we can't target those things. That's why we go through that. And we study that. So purpose of this process and studying it is they are always looking, pharmaceuticals and their research, they are always trying to find little tiny differences so they can come up with better ways of helping your immune system to get rid of it. Your immune system is the best way that can distinguish between self and non-self. But sometimes it needs time to get the whole process, you will see, to get the process started, create the antibodies and all of those things to specifically targets the bacteria or the virus and all of that and not target you. But sometimes it needs a little bit of time to catch up with this process. And that's what the drugs do, right? If you think about antimicrobial drugs specifically, which is chapter 10, we have a lot of different antibiotics. Antibiotics affect bacteria. But when it comes to viruses, fungi, protozoans, we are very limited because a virus uses us. So how can you tell a chemical, which the medicine is, hey, target this cell, leave this other cell that is identical to it alone? The chemical doesn't know infected, not infected. Protozoans, again, same, very similar to us. Fungi, very similar to us. So it's very hard. So we're going to look at antimicrobial reagents. So first, chapter 9 deals with what if the pathogen is outside. So you have a patient who came through and they had measles and you want to clean the area so next patients that come there, they don't get it. TB, so on and so forth. This is outside of body. Chapter 10 talks about antimicrobial drugs. So what is inside? Once the infection is inside, what can you do to help the immune system? And then chapter 11 is a little bit of epidemiology. We go through it and look at like difference between something that gets everyone sick versus something that is a true pathogen versus something that is opportunistic. Our interactions, we'll talk about that and a little bit of epidemiology. And then we'll talk chapter 12 and 13. It's going to be like, okay, how does your immune system react to pathogens? And then we're done with this portion, and the last third of the portion of the class is going to be diseases. All right? Okay, chapter 9. So let's see about controlling growth. Why we have controlling microbial growth? Why don't I use the term death? Because if you can control the growth, even if you, first of all, if you stop the reproduction and growth, that's it, you're done. That bacteria, for example, cannot replicate in numbers. If it cannot replicate, then eventually itself will die and will be gone. But if it keeps replicating, you get rid of one, there's 2 more ahead of it, 2, 4 more, and all so on and so forth. So that's why we keep, sometimes we don't even have to totally destroy everything. If we stop reproduction, we're good, especially for internal infections. That's the part that your immune system needs help with. Immune system eventually will go find that thing, unless your immune system is not functioning, we'll go and we'll say, you are not supposed to be here, and we'll remove that bacteria. But it needs a little bit of time to figure out what the bacteria, the virus, and all of that is. And by stopping the reproduction, that's how you buy it time. So the numbers are not increasing, and it will get a chance to destroy it. Okay, some terms that you might have heard, depending on where you're working with. So let's go through those. Sterilization means removal of all microbes, whether it's a pathogen or not. When you sterilize something, you are removing hopefully majority of microbes. Microorganisms are removed. Proper, like autoclaving, incinerating of your loops. You're using that extreme heat, you're killing anything, any cell, anything that is there, you're melting the proteins, you're destroying. That's sterilization. Okay. Disinfection and disinfectants. Disinfectants are reagents that disinfect. The disinfection means you're removing the important microbes, the pathogenic microbes. Your purpose is not, okay, I'm going to destroy everything. Your purpose is, I'm going to clean this area good enough that, you know, a person with a strong immune system won't be in trouble here. Disinfection and disinfectants typically talk about an object, not a person or living tissue. That's the term we use for objects, inanimate objects. Sanitation or decontamination, usually the term sanitation, like they say, this restaurant is not doing sanitary processes. Sanitation of the public restrooms. That means disinfection of the area for public use. All right, so you're considering pretty much everyone with the different variations of the system. Antisepsis and antiseptic. Antisepsis is basically when you're disinfecting skin, tissue, living organism. So antisepsis is disinfection, but of living material. So that's how we distinguish. Antisepsis we use when you are cleaning a tissue, a living being. Aseptic is an environment or a procedure that is considered to be antiseptic. That it's supposed to be clean. Like a surgery is an aseptic procedure. They create a surgery room that they are hoping that it's clean. People walk in with sterilized gloves, masks, things like that. The bed is sterilized. All the tools have been autoclaved. So they are cleaning the area as best as they can. Is it going to be sterile? No. For a sterile environment, you have to go beyond what they do in surgery rooms. Like the person entirely has to be covered. The room is clean in certain ways. Now, degerming is when you're physically removing a microbe, filtering a solution to get like the bacteria on top of the filter so the rest of the liquid that goes through is clean. That's degerming. So in the water sanitation, that's what they do. It can be for humans, for our tissue, when you use the alcohol swab. The alcohol itself is a disinfectant. It's actually very good. It can almost get to sterilization. But the cotton swab that you rub on the person, what that cotton swab does, it removes the dead skins. So that's degerming and aseptic techniques together. So antisepsis, or antiseptic alcohol with degerming. When the surgeons go scrub, they are using the soap, warm water, but then they use the brush to remove the dead skin. So that's degerming with aseptic techniques. Pasteurization. This is when you basically warm up the liquid. Pasteur came up with it, remember, with the fermentation process and the biogenesis. So when you warm up liquids to get rid of some of the pathogens, and then mostly your intention is to get rid of microbes that cause spoiling of the food. Anytime you see a suffix that says stasis or static, so like homeostasis. Stasis means halt, keeping it halted. It's in a static level. So you are not destroying anything like reproduction, you're just halting it. You're slowing it down. You'll see refrigeration is static. All you're doing is you're freezing or keeping that bacteria cold, so it's not in its optimal environment, so it won't reproduce. But the second you put it in a perfect environment, it's going to regrow and do all of that. Whereas side or sidal means you're completely destroying it. Not only you possibly destroying the original organism, but you completely have, or you can actually, by complete destruction of reproduction, for example, that bacteria, even if it's not dead, it won't be able to reproduce. After 20 minutes, it will try to reproduce, it can't, it will die. All right. So microbial death is permanent loss of reproduction. So when it comes to microbiology, when you're saying you're killing an organism, you are preventing it from reproducing. That's sufficient for us. Okay, so we're going to look at antimicrobial reagents, and we're going to look at it in basically first I'm going to mention about how do they affect the microbes. How can they change their reproduction rate? They can change the reproduction rate by going two different ways. It can be structural. So if you damage the cell wall or prevent them from forming a cell wall, that bacteria, for example, cannot reproduce. If it cannot make new cell wall or cell membrane, it cannot reproduce. Then we have like cytoplasmic, so structural damage. or you can interfere with protein synthesis or nucleic acid reproduction. If it cannot produce proteins, if it cannot reproduce its nucleic acid, it won't be able to reproduce and process. All right. I'll go through the processes as I go through each of them. Relative susceptibility. So all these different microbes that we have. So we have bacteria. In bacteria, we have gram-positive, gram-negative. Then we have mycobacteria, which is acid-fast. They not only have the thick peptidoglycan, but they also have a very thick mycolic acid, right? So that is different kinds of bacteria we have there. Then some of these bacteria can produce endospores. Remember, endospore is something that is very hardy. The bacteria actually produces it to survive very harsh environments. Then you have viruses. You have enveloped virus versus a naked virus. Then you have funguses. Funguses, they are the vegetative form. Vegetative form, when you say like a vegetative form of bacteria or a fungus, that's like when it's growing normally, all right? And you can have the vegetative form of fungus or the fungus spore. The spore is a little bit hardier than the vegetative. Same thing with protozoans. Some of them, they reproduce and they make cysts. Those cysts have a very thick shell. So again, it goes there. So they have a different degree of how hard it is to get rid of them. And that's what we talk about susceptibility rate. Susceptible means how they're affected. Resistant means they are fighting with everything. By the way, if you guys are very hot, I can move you guys to another room that it's cooler. Everyone's okay? All right. Let me know if it gets uncomfortable. All right, so enveloped viruses actually are the most susceptible. It's the easiest thing to get rid of because they have that envelope on the outside, and that's where the spike is. Warm water and soap removes the soap, interacts, you'll see, interacts with the plasma membrane, it interacts with the phospholipid and the different parts that are charged of the phospholipids. It separates them, so it doesn't allow them to stick together. And it actually dissolves lipids. Alcohol very easily removes the envelope. So as long as the envelope is damaged, that virus is incapable of getting into the host. All right? Then we have gram-positive. All you have to do is get rid of the peptidoglycan. The non-enveloped viruses, they have the protein capsid, fungi, gram-negatives, active stage of protozoans, cyst of protozoans, mycobacterium. So this is where TB is, the acid-fast. And then we have bacterial spores, and finally prions. So prions, the only way that we believe we can get rid of prion is complete incineration, extreme heat. Now the reagents that I'm going to show you guys, like for example for the chemicals, mostly we use this for chemicals. We can range them from high level, intermediate level, and low level. That means what groups can they take care of. High level antimicrobials usually get rid of pretty much most things, and they can even get rid of endospores, mycobacteriums. Prions, no, because prions right now we say incineration. Intermediate level is most pathogens, some of the spores, some of the cysts, so they get rid of a lot of things. Low level is just the vegetative states. As far as like where would you use this, high level would be in a place that you really have to have a sterile environment and things like that. Hospitals typically go with intermediate level. Unless they find something like a person comes and they've been infected with TB, those rooms are treated totally differently because TB is very hard to get rid of. And many of them are resistant. So, but most cases, doctors' offices, dental offices, hospitals, we go with intermediate. At home, we're dealing with low level. Really, Yes, a person might get sick in their own house, but at a hospital you have different infections, different patients, different microbes. At home, one person gets sick, then they get better, and you just need to get rid of that, right? All right. Okay, so we're gonna look at antimicrobial agents. We're gonna look at different methods where we create a different physical environment. So we can get rid of microbes by interfering with their optimal environment. That's the physical part. And then we can have some chemicals. First one, heat. Changing the temperature and using extreme or higher temperatures has been used by humans for centuries. Heat is a fantastic way of getting rid of microbes or getting rid of anything by killing it using heat. And don't focus so much on it, but like when they are looking at the effect of heat for antimicrobial purposes, they can actually look at it in two ways. Thermal death point is basically, they say, okay, in 10 minutes, what is the lowest temperature I can get to that would get rid of this microbe? So they're looking for the lowest temperature that after 10 minutes at that temperature, it gets rid of that microbe, stops the reproduction. That death point, so you're looking at a temperature point. Thermal death time is you're looking at a duration. So you choose a specific temperature. Let's say you are trying to clean a plastic tube and you know based on the manufacturing protocols, this tube cannot, the temperature is safe, the tube is safe if you just increase the temperature to 50 degrees. Beyond that, you're gonna damage the tube. So you're gonna see how long do you have to keep it at 50 degrees for you to ensure that this thing is clean to the level that you want. So that's death time. You also, when you're looking at heat, you're looking at dry heat versus moist heat. Dry heat is your oven, moist heat is steam or water.